Hello, and welcome back to day 2 of Advances in Proteomics and Metabolomics online symposium. I'm Elizabeth Williams, conference producer at Technology Networks, and I'm excited to introduce our opening talk of day 2 with Dr. Simon Fredriksson from Pixelgen Technologies presenting single-cell protein interactomes by the proximity network assay. And I am very pleased to welcome Simon Fredriksson. Simon is continually fascinated by technological innovation that pushes the frontiers of scientific discovery into new and uncharted territories. His deep experience as a life science entrepreneur, researcher, and inventor has spanned from genomics to proteomics and research tools. Simon was co-founder, chief science officer, and CEI of Olink AB, which later became Olink Proteomics AB, a publicly traded proteomics company now acquired by Thermo Fisher, where he led the development and commercialization of multiple product lines, most notably the Proximity Extension Assays, PEA, platform for high-throughput multiplex protein detection. A very warm welcome to you, Simon. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And with that, I will now hand over to Simon to begin his presentation.
Thank you for having me. It's a pleasure to be here. So we're going to talk about proximity assays for scalable proteomics, from blood proteins to nanoscale spatial protein interactome organizations of single cells, using various versions of proximity DNA-based methods, such as proximity ligation assay, and as mentioned already, the proximity extension assay, and now leading up to what we're up to at Pixelgen, which is the proximity network assay. So the introduction of the DNA proximity assays was done over 20 years ago in a publication I was part of when doing my PhD work in 2002 in Nature Biotechnology. So the proximity ligation assay then made use of DNA aptamers that bound to the protein platelet-derived growth factor BB. And the idea behind this was that, if we could have dual recognition of proteins, which ensures specificity, and generate a DNA-based readout by the combination of two different DNA sequences, we could get specificity, sensitivity, and throughput in plasma-based proteomics, and eventually build this out into a highly multiplexed method. But it all starts with one, and that's what we published in 2002, and introduced the concept of proximity assays for protein detection, which is now a household tool. Later on, we figured out how to actually couple DNA in a very high quality way to antibodies. And of course, antibodies are very generally available as matched pairs or polyclonals to detect many proteins. So once we had that figured out, we could use proximity ligation to detect now cytokines for almost any cytokine that there was a antibody pair for. And we gained very good sensitivity. There was also a very minute sample consumption. And the readout was always with the newly at that time introduced time PCR to get a good throughput of the assay, but still only in singleplex. But the dream was to build this out into a highly multiplexed method, because there's a scalability bottleneck in plasma proteomics. So if you want to detect a lot of proteins in a plasma at the same time using many sandwich immunoassays, they don't really scale that well because the antibodies are not perfect. And the more antibodies you throw into one and the same reaction start cross-reacting, because the probability of cross-reacting antibodies increases exponentially with the number of assays tried to do. That's where many assays at the time, Luminex and others, were limited to about a 20-plex assay. So what we theoretically speculated was that could then, with sequence-based recognition of each unique antibody pair, working pairwise, that sequence 1A would be united with 1B for protein 1, and so on. going forward. This would give us the specificity to really be able to scale plasma-based discovery proteomics in a completely new way. So moving on to Stanford University, we developed the multiplex proximity ligation assay and published that in 2007. And just about a couple of years earlier, we had formally founded the company Olink AB back in Uppsala University, where this method originates in the lab of Ulf Landegren. And this work here that we did in multiplexing, we applied it to a few early studies where we measured proteins in about 30 samples, so 7 proteins in 30 samples, with a multiplexed version of the method, still read by real-time PCR. But we had really good sensitivity, specificity. We proved that we could make affinity proteomics as a tool for high-throughput biomarker discovery. We applied it to pancreatic cancer and ovarian cancer detection and tried to build the first types of combined signatures for detection of cancer. So moving on to Olink, I moved back to Uppsala, where Olink resides, and we developed some proximity extension assay. And that's because the proximity ligation assay, which uses a DNA ligase, it had some inhibitory effects of the nature of plasma interference. But when we switched the method from a ligase to a polymerase, hence the new name proximity extension assay, this was no longer the case. So we could start scaling the method with really, really great specificity across any sample type. So it wasn't dependent on the particular sample that we ran. So we published that in 2011, showing scalability. And in 2013, we published a 96-plex assay with excellent scalability, specificity, sensitivity. So we got this exact same result if we measured 24 proteins or 96 proteins. Still rather low numbers, but we'll get to that. And we launched a product on this that was read on the Fluidigm Biomark high-throughput qPCR instrument. So you ran the assay, you generated your PCR amplicons that were then fed into the Fluidigm Biomark instrument, which ran 96 by 96 real-time PCR reactions. And this was done in March 2013. And we continued to develop more and more panels to the research market on this method so that people could start accessing biobanked blood plasma samples, thaw just 1 microliter of these, and run very, very large, for the time being, biomarker studies. And the effect of the scalability of chemistry is really astounding. Other companies and techniques tried to scale their methods with real estate, building more and more bigger and more elaborate assays. But we chose this way of scaling by chemistry and making sure that the assays are still specific even when you're running them at high multiplexed. So I calculated that we've followed Moore's law from the year 2002, where we published a 1-plex assay, and then leading on to a 6-plex assay in 2007, moving to 24, 96. And a few years ago, a 384-plex assay was released by Olink in the year 2020. And if that continues on, they should be hitting about 1,500 quite soon. I hope that's in the works. Another great thing is that, once we had the scalability of the chemistry, we could develop very many panels and apply this broadly to research. And there are now about 3,000 PEA publications that have used this. It's a household tool in most pharmaceutical companies. And there are now 5,400 protein assays available from Olink. And this scalability of the chemistry has enabled population scale proteomics, as exemplified by the UK Biobank project, where 54,000 have been analyzed for 3,000 proteins so far by Olink's proximity extension assay. So if you think back to the year 2006-07, where we assayed 7 proteins in 30 samples and thought that was really great, translating that into 54,000 samples for 3,000 proteins is an incredible achievement due to the scalability. So in these larger cohort studies, disease prediction by plasma proteomics prior to symptoms has been enabled in pilot studies on dementia and multiple cancers and so on. And this has led to a lot of landmark publications in Nature around these proteomic profiles predicting future dementia and possible cancer cases as well. And earlier this year, or was it last year, they announced that phase 2 is now ongoing of this study for 600,000, 5,400 proteins, furthering the scalability of the chemistry even to the next level. So these types of techniques will then revolutionize the way we study proteomics in blood and lead to diagnostics as well. So these proximity-based chemistries for plasma proteomics is gaining a lot of adoption. There is one, as far as I know, on the market, the diagnostic product by Octave Bioscience for patient monitoring in multiple sclerosis, MS, by proximity extension assay. And this approach of using DNA proximity to gain scalability in plasma proteomics is not only used by Olink and Thermo Fisher, but also Alamar Biosciences, Nomic, and Spear Bio. So it's gaining more use across multiple companies as well. But I'd like to shift gears now to what we're doing at Pixelgen. And diseases are caused by inherent cell. So moving from plasma proteomics to cellular proteomics now. You can see the various omics fields as DNA being a blueprint and RNA being a manufacturing message, and proteins are driving cellular functions. But there is something that is missing in this picture because proteins never work alone, and they work by interacting with other proteins. So the function of a cell emerges from the organization of the proteins, which you can't see if you just measure the abundance. So cell functions are executed by proteins, and they don't execute those functions but in complexes with other proteins. And this is something we're beginning to call protein interactomics. So this is the true nature of which cells execute their systems. And most of the research that we're doing across the world is, trying to figure out what's the difference between a healthy and a diseased cell, and what could we do about it? Can we make better diagnostics or better medicines to modulate these functions? So protein interactomics is the next level of omics that's going to be crucial for driving our understanding and ability to treat diseases. I like to view these as a technology shift. So if you think about the various technology shifts that has occurred over the decades, is that you have one technology usually to make the early discoveries, but then when it comes to translating those discoveries into clinical research, a new type of technology is necessary, driving the adoption of larger studies and eventually diagnostics. So if you think of the human genome, it was first sequenced by Sanger sequencing, which is, of course, very, very slow compared to the technologies that we have today. So phase 1 uses slow technology at low throughput to build the first map, right? But now we have next generation sequencing that can generate an enormous amount of genomes over a year, thanks to companies like Illumina, Ultima Element, MGI, and so on. This has happened in the other omics fields as well. In transcriptomics, the RNA, which is a message, it's a bulk parts lists per cell. Usually studied by serial analysis of gene expression and Sanger sequencing just to figure out, all right, which RNAs actually are inside cells. But when it comes to tracking that into then a clinical research, we need DNA microarrays from Affymetrix and Agilent and so on. And by next generation sequencing from Illumina or single cell RNA-seq from Fluidigm and 10x Genomics. So that those technology shifts enable these translational research settings. The same as that happened with proteomics, as we talked about here, where these components and bulk parts of cells can be analyzed with mass spectrometry, once again discovering which proteins are actually there. But once you want to study hundreds to thousands, now hundreds of thousands of samples, mass spectrometry does not have the throughput or sensitivity required. So therefore, plasma proteomics enabled by Olink, SomaLogic, and other companies enable that. The same thing with single cell inflammatory and similar applications. And usually though, these methods are targeted for proteomics because we need antibodies or other recognition elements to actually find and recognize the proteins. So what about this next omics field we discussed, protein interactomics? So these are the real functional modules driving cell activity and executing cell function. So you can have discovery technologies with mass spectrometry and pull downs, used to hybrid screens, or even AlphaFold, figuring out which proteins may or may not be interacting with each other just based on structure. And these databases are getting better and better. So the second phase of this is move into the translational research again. And here we have PixelGen delivering a new technique that we call proximity networks for single cell protein interactomics. We can measure in a targeted way multiple protein interactions on the surface of a cell. We're going to spend the next 20, 30 minutes talking about that. So thinking about the fact that, cellular function is driven by protein interactions, this is the next focus of research. And the protein organization is critical for many different cell functions. And here are a few examples. A migration, a resting cell versus a migrating immune cell will have a different shape and a different protein organization, forming a front and a back end. And having immune cells wander into a tumor is the most crucial aspect of having a positive outcome in any oncology case, irrespective of treatment. And the mechanism that happens by is by migration. Mechanism of action of drugs, many drugs bind to target proteins on the surface of various immune cells or cancer cells and modulate their activity or cluster them together. When cells interact with one another, like a CAR T in a tumor cell, you have a cell-cell interaction that's then relieved and the cancer cell is killed. And a process called trogocytosis leaves a patch of the cancer cell on the CAR T cell. And this is, of course, a really crucial event with protein organization driving the cell-cell interaction, both pre and post the actual cell contact. And tonic signaling can occur if a cell surface receptor is highly clustered, and it can exhaust immune cells, for example. So all of these very essential functions to immunology are driven by spatial reorganizations and protein interactions on the surface of immune cells. So what we've delivered at PixelGen is what we call the PixelGen Proxiome kit based on the proximity network assay. It's a very simple kit-based product that takes single cells in suspension or cell-cell complexes. You run our assay and you sequence it, and then the sequencing file is processed by a software, a freeware open source software, to give you three-dimensional reconstructions of single cells. And the software then figures out where the proteins are in spatial relationship to each other in a proximity-based assay that makes multiple connections. So we're going to watch a little video to see how that chemistry works, because it's easiest explained in an animation here.
The proximity network assay workflow starts with cells in suspension being fixed using PFA to immobilize the proteins on its surface. In the next step, DNA bar-coded antibodies are added to the mixture and bind to their complementary protein targets on the cell surface. This is followed by the addition of padlock probes, initiating a rolling circle amplification reaction, generating an amplification product on top of the antibody at very high resolution. Next, linker oligos and enzymes are added, and a gap-fill ligation reaction creates a DNA fragment containing barcodes for proteins in nanoscale proximity, forming a network encompassing each single cell. After sample index PCR, the DNA fragment contains both sample indices and sequencing adapters. The DNA fragments are processed through next-generation sequencing, and the NGS output data is processed in our Pixelgen software to form a high-resolution graph network of the proteins of the cell surface, with direct connections between neighboring proteins. The proximity network assay produces single-cell data which is 3D in nature, showing a representation of the cell surface. Over 150 protein targets are assayed in parallel, and scores for protein clustering, colocalization, and abundance are generated per single cell. Up to 50,000 unique proteins are detected per cell. This high-resolution protein interactome information will lead to advancements in translational hematology, cell therapy, immuno-oncology research, and beyond. This is Proximity Networks, life at the highest resolution.
There, thank you for viewing that. So the way the proximity network assay works is that it's very similar to proximity ligation and proximity extension, but each oligo tag on the antibody is first copied a few times by rolling circle amplification, which is a local amplification reaction. That enables you to make multiple connections to nearby antibody oligonucleotide conjugates or barcoded antibodies. And if you can make multiple connections to nearby neighbors, you form a network. And that is the basis of building this sprawling proximity network across the entire cells. So first, PFA-fixed cells are bound by the barcoded antibodies. You do the rolling circle replication with the padlock probes. And then the linker oligos unite the proximal antibodies, forming the spatial network. So if we compare proximity networks to other proteomic analysis of cellular function, there are multiple techniques that give you a very good measurement of the abundance of proteins on the surface of single cells, like flow cytometry, mass cytometry, or CytSeq with antibody DNA tags, but they have no spatial resolution. So if you want spatial resolution, you need a microscope of some kind. A regular microscope can give you 3 or 4 plex per run at 200 to 500 nanometers resolution, or a super resolution microscope, which could do 1 or 2 plex, but at very high resolution, like 20 to 50 nanometers. But all the microscopy methods, they usually give you 2-dimensional data for a few cells at a time, very low multiplex, because of the limitations of light, that you can't use an infinite number of colors at the same time. So the proximity network assay fills this upper quadrant with very high resolution at 50 nanometers with a 155 plex kit that we've launched so far, with the data naturally in 3D. So it's the equivalent of taking a picture of the cell from all angles at the same time, always in focus, because it's a chemical assay. And this is a very, very powerful new approach that people are calling DNA microscopy. So you have high multiplex, you have spatial information of about 50,000 localized protein molecules per cell. And the way we've estimated the resolution of the method is that we take the surface area of a lymphocyte, divide it by 50,000 spatial positions, and we get an estimated resolution on average around 50 nanometers there. The method has high throughput. We can analyze 1,000 cells per sample at 8 samples at a time. And it has a very interesting development trajectory, as it's only limited by sequencing capacity, computational power, and so on going forward. So we have a scalable way to relieve the shortcomings of microscopy. And the microscope was invented 350 years ago and has taught us almost everything we know about cellular life. But now there's a new method, the proximity network assay, that can tell us an enormous amount of information about the mechanisms driving cell disease. So in this Proxiome kit protein list, we have a collection of 155 proteins spanning adhesion proteins, co-stimulatory molecules, T-cell receptor components, FC receptors, and so on. to really capture a very broad research space. And the data output from this proximity network assay, since this is a new data type, it's not just giving you, this is one protein, here's the amount of it, but it has this network effect, these massive networks of proteins interacting with each other. So first of all, of course, we also get the abundance of each protein that's detected on each single cell. So if there's a low abundance, of course, that's low, and higher abundance, it's higher. And then we've developed two spatial metrics, which provides the novelty of our method. And the clustering is, of course, then how dispersed is one protein versus how clustered is it with itself. So the clustering score would go up if it has a higher value. And then the last one is colocalization metric. So we walk pairwise through each single cell to determine if two proteins are segregated away from each other, which would give a negative score, if they're randomly colocalized, or have a positive colocalization, more likely to be interacting with each other. And these spatial data sets are normalized by the predicted randomness. If you have two, for example, very high abundant proteins, they are likely to be close to each other in the network. But we compare our scores with what's predicted by random occurrence to give you a statistically significant result that's not just driven by random colocalization driven by abundance, which almost all other protein interactomics techniques are hindered by. And the fact that we have a single cell technology enables us to study clinical samples. So this is really an important point here. Protein interactomics has been done on cell lines where you have tens of millions of cells and mass spectrometry or microscopy. But now we can first, using protein abundance, annotate our cell types in a PBMC sample in a classical way to find your platelets, monocytes, B cells, and so on. But going forward, each cell in this sample then has a protein distribution. This is CD45 on a naive CD4 T cell. And you get the abundance measurements down here for all these proteins, and the data are highly validated and correlate well with the flow cytometry or other standard techniques. So when it comes to protein clustering, one element of clustering is that drugs like rituximab pulls CD20 all to one end of a B cell cancer, triggering NK cell activity. And this is clearly seen by our protein clustering score, that CD20 goes to one end and drives the clustering score, which is also seen by traditional microscopy. But the level of data that we can generate with proximity networks is, of course, much, much higher than you can generate with a microscope. So protein colocalization, segregation versus colocalization here. You have a lot of protein complexes, multi-protein domains on the surface of cells driving their functions. And these are clearly seen in our data sets. You can even look for well-known protein interactions, HLA-ABC, B2M, CD11A, CD18 forming the LFA1 or the VLA4 complex of CD20, CD49D. As these are popping out as more colocalized than predicted by random. And the data has also been validated with super resolution microscopy with using standard fluorescence-based colocalization. So we have a preprint of this method available that is currently under review. And if you look then at a colocalization map of this cell here, you see a lot of these CD45 isoforms, HLA, ABC, B2M complexes, and so on appearing. And you can compare microscopy images of CD40 with ICAM1, which is more at one end of this cell, and CD81, which is very patchy, and compare that to the proximity scores that we have of our method. And below here in the bottom panel, we have the graph networks of each cell example of this data set. So another few examples of what this can be used for is to study cell migration. So when immune cells migrate, they will form the uropod end, a front end, and a back end. And this can be enabled in vitro by adding cytokines or chemokines, I should say, RANTES, MCP1, on an ICAM1 coated surface where the T cells are resting. They then form a uropod that sticks out from the cell, which we see in our data for CD50 here, and which is also seen in the microscopy images here. So this gives you a high throughput way to study one of the most important mechanisms of immune cells, which is their migratory capacity to find cancer cells, to find infected cells, and so on. When it comes to larger studies for biomarkers, we've done a few work in hematological oncology, acute myeloid leukemia, trying to find which patients will respond and who will not respond to a certain drug. So in this case, we studied together with Aaron Vinnie at Columbia, AML patients responding or not responding to hypomethylating agents. So we built a random forest classifier looking at AML blast cells and other cell types from the bone marrow. And we could then build in this pilot study a predictor with 94% accuracy based on not only just abundance, but clustering and colocalization of multiple protein interaction markers to increase the predictability of who will respond to these types of drugs. In immune therapy, the protein interactome of CAR T cells is really essential to how they function. And we've studied CAR T cells, also having a probe that locates where the anti-CD19 CAR of this particular CAR T cell is. So in a mixture of cells, you can find the CAR T cell and which proteins are close to it and compare various constructs with each other. And for example, do co-culture studies with CAR T cells and Raji cancer cells, which over time will then form complexes and kill the Raji cells. This drives massive changes in the spatial organization of proteins, but also in the abundance of various proteins, which is seen and exemplified here, driving up certain markers over time and others becoming downregulated. But the most interesting part of this story is the spatial organization of CAR T cells after they have encountered their cancer cells. So after they bound cancer cells, we found that ICAM1, CD54, is very abundant on the CAR T cells, but this is a B cell marker. So these B cell markers form clusters on the surface of the CAR T cell. And there's multiple B cell markers here, like CD40, ICAM1. And this is through the process of trogocytosis. When a CAR T cell binds a tumor cell, it forms a cell-cell interaction and then leaves a patch of cancer cell material on the tumor cell. This can be quantified with our software to figure out that, how many patches there are per single cell, and correlated to various exhaustion markers like PD-1 or activation markers CD25, giving you new insights into how CAR Ts function. We did a small biomarker study in lupus. So this is an autoimmune disease driven by B cells. And CD21 is a protein of genetic predisposition to SLE. And we found that the abundance of CD21 in SLE cases goes down compared to healthy controls, but it also clusters more, leading to higher colocalization of CD21 with itself, perhaps driving its regulation of the B cell receptor. So this gives you new insights into potential mechanisms behind severe diseases of autoimmune type, such as SLE. So this is just a small snippet of information from a very large study with lots and lots of data behind it. So to summarize, we can apply this Proxiome kit based on the proximity network assays to hematological oncology, immune therapy, cell engineering, basic immunology. Autoimmunity is a huge field, also immuno-oncology and drug development. But I'll stop there. I'm very happy to take any questions.
Thank you very much, Simon, for a really fascinating and informative talk. I can see we've received some questions already, so I'll move straight on to the Q&A now. First question, could you expand on how the proximity network assay achieves nanoscale resolution without the use of optics?
Right. So the proximity network assay works only with DNA and not light and fluorophores. So using DNA tagged antibodies, each target protein is then identified by the barcoded antibody. The DNA tag is then amplified quickly for a few minutes by rolling circle amplification, enabling it to make multiple contact points to nearby neighbors in something that people are now referring to as a DNA microscopy. So instead of light microscopy, fluorescence microscopy, atomic force microscopy, this new way of localizing biomolecules by DNA is spearheading a new high throughput omics type of research.
Wonderful, thank you. And another question, how does PNA compare in terms of sensitivity and resolution to existing proximity ligation assays or imaging-based approaches like super resolution microscopy?
Yeah, so in situ proximity ligation assay, which is a version of proximity ligation assay that generates a fluorescent signal inside tissues and cells to look at protein interactions, for example. It has an estimated resolution of around 40 to 50 nanometers, which is the same as our estimated resolution on the proximity network assay. On the other hand, we make a sprawling network of potentially 155 by 155 protein interactions. So it's more of an omics tool since it does not rely on the use of light and microscopes. And when it comes to the spatial resolution of super resolution microscopy, which I believe can come down all the way to 10 nanometers, that is, of course, higher than our DNA-based methods because we require an antibody and the DNA oligo itself does occupy some space, making it a little less highly resolved than super resolution microscopy, but on the other hand, with massive throughput to generate hypotheses that can be verified with super resolution microscopy.
Thank you. In your examples, how did proximity network assay reveal new insights into hematological oncology, autoimmune disease, or CAR T therapy that might not have been detectable with other methods?
Yeah, so modern research is done in more or less hypothesis-free way with new technologies driving them. So instead of asking a question, does CD21 cluster on the surface of B cells and assaying that with microscopy, you don't have to have a clear research question going into your biomarker study. So you cast a broad net as broad as possible with as many proteins as possible. And then you discover things that you may or may not have been looking for, like the CD21 clustering on severe lupus cases is very, very mechanistically interesting and would not have been, at least by me or other researchers in the field, predicted. And there's, of course, much more data in that data set. So that gives us new clues to what's driving these mechanisms. And we will continue to expand on our offering from 155 proteins, going much, much further with this scalable chemistry. So that's one example.
Wonderful, thank you. And another question, how might PNA be applied to study dynamic processes, essential for cell function, such as migration attachment, where proteins interactions are highly transient?
Yeah, so that's a great question. So the proximity network assay works on PFA fixed cells. So all the proteins are locked into place throughout the assay method. So we don't study the real-time dynamics of protein interactions. But on the other hand, you can start one reaction and take time points from it to study that. So and then look at the dynamic changes over time in an in vitro co-culture of cells or a stimulation of cells and so on. But we do not measure that in real time on one and the same cell.
Thank you, that's clear. Are there plans to integrate PNA with other single-cell omics technologies, such as transcriptomics or epigenomics, to provide a more complete system-level view?
Yeah, I mean, the proximity network assay is very versatile. You essentially do rolling circle replication reactions, and you unite those sequences with whomever is nearby. Those rolling circle replication reactions in our kit today are generated from proteins, but they could be RNA molecules as well, or other types of molecules that you could tag with a little piece of DNA. So there's not really any limitations there. So likely that will happen in the future.
Wonderful. Thank you so much, Simon. Unfortunately, that's all the questions we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all of the other talks in this event, the talks will now be available to view whenever you want to for the next 30 days. That just leaves me to say thank you so much again, Simon, and we'll be back soon with our next talk.
Thank you.